Okay, in the last section we talked about proteins. In this one we're going to talk about nucleic acids. And what nucleic acids basically do is they store the hereditary information. That's the information that we know as our genes, and that's the information that is basically going to be transcribed and then translated into protein. And we'll talk about transcription and translation later on in the course. There's two main types of nucleic acids. You have DNA and you have RNA. The DNA provides the necessary information for constructing the proteins. It gives rise to RNA, which is then transferred to the ribosome, which is where the proteins are made. And it also contains the information that's necessary for guiding its own replication. And we'll kind of go through just a brief example here, and we'll talk a little bit more about this in class as well. What the DNA can also do is it can also guide the synthesis of RNA, and during the process of transcription we generate this messenger RNA, which is can be modified a little bit and then sent out to the ribosomes, or in the case of a prokaryotic cell, it can give rise to protein right away. But either way, the nucleic acid, what that's going to do, the DNA especially, is going to contain the information that's necessary for making protein. Okay, so this flow of information that we've been talking about from DNA to RNA to protein, that makes up what's known as the central dogma of biology. In other words, the information that's used to create the proteins is contained within the DNA. And in order to get that protein, we've got to synthesize this messenger RNA from that DNA. And then those proteins are basically what makes us who we are. They're responsible for running the cell. They give us all of the different features that we have. And really the main difference in the way we all look, whether it's dark hair or light hair, dark skin, light skin, those types of things, is just only a few nucleotide differences in the actual DNA molecule, those A's, T's, C's, and G's that you've probably learned about in an earlier biology class. Very few difference in those is what gives rise to all of the different features that we have as human beings, because those slight differences make a big difference in the protein and how much of it is made, and so on and so forth. Okay? So if you look here, this is basically showing you that central dogma in a picture. You can see in the nucleus, you've got the DNA there in blue, the double helix, double-stranded molecule. That gets synthesized into the red ribbon there, which is messenger RNA. That occurs inside the nucleus, and then it gets out of the nucleus, and that messenger RNA in the cytoplasm then travels to a ribosome, which is where it's used to make proteins. And there's some more details that we'll cover as the course goes on, but this is just detailing, or I guess I should say simplifying, what's going on in a eukaryotic cell. The same type of process occurs in a prokaryotic cell, except of course there's no nucleus. The DNA is transcribed into messenger RNA, and then that's immediately available for use. It doesn't have to travel anywhere other than to the ribosome. Okay? So nucleic acids, they're basically referred to as polynucleotides. They're polymers, of course, because the individual monomers are called nucleotides. And what the nucleotide has on it is, you can see it's got a sugar, a phosphate, and a nitrogenous base. And you can see that in the box there on the right. And what the nucleotides are composed of is the sugar, the phosphate, and the base. If you hear the word nucleoside, all that is that's a sugar and the base. Okay, and we went over this a little bit in class in terms of how these things get stacked up to give rise to the single strand of the DNA molecule. Okay, so the nucleotide monomers, of course, these are built from the a sugar and the nitrogenous base, and then we add the phosphate to it. And you can see the different types of nitrogenous bases here. You've got the purines, which are the adenine and the guanine. And then up above in that box there, you've got cytosine, thymine, and uracil being found in RNA. And then those are the pyrimidines. Those are what get joined to the sugars down below. In the box below, you have two pentose sugars. Pentose, of course, is a 5-carbon sugar. You have ribose, which is on the right, and you have deoxyribose, which is on the left. And the only difference between those two sugars, if you look at the number 2 carbon in ribose on the right there, the number 2 carbon has a hydroxyl group on it. And if you look at the deoxyribose there on the left, the number 2 carbon is missing the oxygen, hence the term deoxy. So they're both essentially the same. They're both pentose sugars, 5-carbon sugars. The only difference is the existence of the hydroxyl group on the ribose. Those join with either a purine or a pyrimidine to give you the nucleoside, and then you add that phosphate group, which makes it into the nucleotide. The nucleotides get linked together now by these phosphodiester bonds, between a hydroxyl group on the 3' carbon on one nucleotide and the 5' carbon on the next. So if we look here at this figure, I'll go back in a sec. If we look at this figure here, you can see the number 3 in red on the right figure, and you can see the number 5 in red on the right figure here. And that the number 5 carbon and the number 3 carbon, that's where you're going to have a joining of that phosphate group. Okay, so the figure on the left there basically shows you how this phosphate group is attached. So you can see it's attached to the 5' carbon there. And then as they get attached, that's what's going to give rise to this sugar phosphate bonds, and that's going to give rise to the backbone of the DNA. Okay, in the sequence of these things now, if we look here again, you can see this on the figure on the right, it goes T, A, that's thymine, adenine, adenine. Those are the nucleotides that are getting stacked on that base as it's being built, and that's what's giving rise to the DNA, the genetic code, and so on and so forth. And we'll go through more of this type of stuff in class, and we'll get into more details of this, of course, as the class wears on. So if you have any questions, of course, make sure you write them down. I'll be sure to go over them with you in class. Now, we've talked about DNA. RNA is a single strand of nucleic acids in this polynucleotide chain. DNA, we said, of course, was double-stranded, and the idea that it's double-stranded, what that means in terms of having complementary base pairs and these things being anti-parallel is that one chain can always give rise to the next. So these two strands of DNA are joined along their length by these sugar phosphate bonds, okay? And then they're joined together side by side. If we've got DNA, we've got the two strands, they get joined together by hydrogen bonds, and then their shape kind of gets spun almost like a ladder that's getting twisted, and then you have some van der Waals interactions between the two strands. And again, I'll go over this more in class as well, but I'm talking about it now because we've talked about hydrogen bonding, we've talked about van der Waals interactions in class, in these things and how they interact with different types of molecules and between water and so forth. But I want you to know that they exist between all sorts of molecules and all sorts of substances. So these types of things aren't going away, which is why we introduced it early. And it plays into kind of holding these molecules together here. So if you look now, you can see the dotted lines between these orange and the blue strands there. Those dots, of course, are the hydrogen bonds. And then along the top portion of this, that's where you have this twisting you can see. And this is just a picture of kind of DNA synthesis. But along that twisting, you have some van der Waals interactions that are holding that together as well. Okay? So the bases that you find in DNA, of course, and we've discussed this, and most of you know A, T, C, and G, adenine, thymine, cytosine, and guanine. Those are found in DNA. And then RNA has A, U, C, and G. Adenine, uracil, thymine, and guanine. So when they pair together, of course, you have adenine pairing with thymine, and you have cytosine pairing with guanine in DNA. And then with RNA, you have uracil in place of the thymine. So adenine is pairing with uracil, and cytosine is pairing with guanine. So A to T and C to G in DNA, A to U and C to G in RNA. Okay? So if you look at these two strands, we talked a moment ago about them being complementary. In other words, one strand can give rise to the next. And the 5' and 3', that will make more sense to you when we talk about DNA synthesis and that sort of thing. But what it's referring to is where the 5' carbon is on the molecules and where the 3' carbon is. And if you were to take these two strands apart, and you can try this, and we're going to go over this in class as well. If you take these two strands apart and just write down, for instance, AGG, TCCG, and then you fill in going back the other way, you'll give rise to what we have here, which would be going the opposite direction, would be CGG, ACCT. And then if you do the same thing with the bottom strand, again, you're always going to be able to give rise to the next. And just know that the 5' end on the top molecule pairs with the 3' end at the bottom. And then if you go over, the 5' on the bottom pairs with the 3' on the top. And I'll go over what that means. It'll make a lot more sense again when we start talking about DNA replication and all of that stuff, in the future. But just know now that we say that these things are complementary. These two strands here are complementary because each one can give rise to the other. Okay? So with nucleic acids now, this is what's going, this complementary nature of the things is what makes the exact copying possible. In other words, that's why when the cells divide, we get clones of these cells in our body, which is what we want. And it's because when the two strands separate, they each serve as a template for the construction of, the two new ones, which hopefully are going to be identical. We don't want any mutations. And now these daughter cells, when they divide, they each contain the complement of that original parent cell. Okay? So the parts of the DNA molecule, which you've told me in class, you've got these triplets, these three bases that are going to code for the particular amino acid. And this information can be used from an evolutionary standpoint to just see how closely things are related. So when we talk about evolution, we're going to kind of come back and review some of this and just discuss kind of what's going on. But what molecular biologists are able to do is they're able to sequence these genes, and they're able to sequence the proteins and things like that, and they're able to kind of look at and see how much difference there is between one organism and the next, and this is what helps them kind of construct the tree of life to look at and see how certain things are related to one another. So for instance, if we're looking at the polypeptide, the protein chain now of the human hemoglobin, and we're comparing it to that of a gorilla because we want to see if they're both closely related or not, you'll find that there's only one amino acid difference out of 146 that are making up the human hemoglobin and the gorilla hemoglobin. So that would suggest to you that the two are very closely related. And then if you look at gibbons, which is another type of a tree monkey, which is very closely related to humans and gorillas, there's only a difference between the hemoglobin of the gibbon and the hemoglobin of the human of 2 amino acids. So some of the information that you could glean from that is just saying that the three are very closely related, and that the human and the gorilla are more closely related than the human and the gibbon because there's only 2 amino acid, or because there's 2 amino acid differences between the human and the gibbon versus only 1 between the human and the gorilla. Okay, so there's other things that can go into account there to see how closely related these organisms are, and it just kind of serves as a starting point. And if you were to look at the DNA, you would find some very similar types of similarities between these 3 types of organisms. Okay, and again, when we talk about evolution and we get into the sequencing and we start talking about cladistics and things like that, we'll kind of revisit these types of things. But I just want to introduce it now. It's an early chapter. It's kind of introductory material, so I just want to make sure that you're understanding what's going on. Okay? So let's watch this video.
In DNA and RNA molecules, nucleotides are strung together in long chains formed by covalent bonds between the sugar of one nucleotide and the phosphate of the next. RNA usually occurs as a single strand, formed in this manner. DNA, on the other hand, is usually found in the nucleus of cells with 2 strands, bound together by hydrogen bonds between the nitrogen bases. However, only adenine and thymine bases and guanine and cytosine bases can bond together. Because of the different chemical bonds and monomers that make up DNA molecules, they take on the appearance of a twisted ladder, or what scientists call a double helix. DNA molecules can be millions of nucleotides long. Each long, complex DNA molecule in the nucleus of a eukaryotic cell is called a chromosome. Chromosomes contain subdivisions called genes. The sequence of nucleotides in most genes contains a kind of biological Morse code that has the instructions for building one complete protein. As we saw earlier, these instructions are copied onto RNA molecules that travel into the cell's cytoplasm and there direct the synthesis of proteins by ribosomes.
Okay, so that wraps up the protein section, the nucleic acid section. So if you have any questions, please make sure you write them down. We'll discuss this a little bit more in class. Okay, we'll see you soon.